Good afternoon, everyone. It's a pleasure to welcome you, those of you here in Missouri Auditorium and people watching by video, to our Wednesday afternoon lecture, which this afternoon is going to be given by Dr. Hoekstra, entitled Digging for Genes That Affect Behavior, and there's a pun in there that will become clear in the course of the presentation. Dr. Hoekstra did her undergraduate work at University of California in Berkeley. I read that she went there not because it was the only place she got in. She got into a lot of places, but she wanted to play Pac-10 volleyball, and I hope that turned out well. And went on from there to get a PhD in zoology at the University of Washington in Seattle. Following a stint at UC San Diego as assistant professor, and before that as a postdoc at the University of Arizona, a postdoc, I might say, which was a Ruth Kirstein NIH NRSA postdoctoral fellow, something we're always proud of when people turn out really well, as she has done. She has been, though, at Harvard for the past, I guess, about 10 years, where she is now the Alexander Agassiz Professor of Zoology and Curator of Mammals in the Museum of Comparative Zoology. None of you can claim to be a curator of mammals, but our speaker has that title, and you will see just how curating she has been with her research. She has a history of being an awesome professor. I read a bit about that. You may get a chance to see her teaching skills here in this lecture. She's an elected member of the National Academy of Sciences this year, and she won the very coveted Richard Lounsbury Award from the National Academy of Sciences last year. Her name has suggested to some people that she must have Native American origins, but I understand that Hopi actually means little bundle in Dutch. The nickname that was given to her by her grandmother and apparently has stuck. So we're in for a treat. So please join me in welcoming Dr. Hoekstra.
First, can everybody hear me okay? Yeah, great. Okay, so it's a great pleasure to be here. I've had a fantastic day meeting with people talking about vasopressin and ADHD and feeding behavior, and it's been wonderful. And the students were great at lunch today. So I've had a very exciting day, and now the opportunity to tell you a little bit about the work that we've been doing over the last several years, trying to make the links between genes and behavior. So my lab, in general, is very interested in understanding the molecular and genetic basis of adaptation, so traits that matter to organisms in their natural environments and increase their ability to survive and ultimately reproduce to pass their genes on to the next generation. Now, initially when we started the lab, most of the work we focused on were morphological adaptations, one of which is highlighted in this image here. This is a mouse that lives on the Gulf Coast of Florida. And what I think you can tell from this image is it has a very light coat relative to a typical mouse. And we've been able to now identify the genes and mutations in genes and the developmental processes through which these genetic changes leads to this morphological adaptation, which gives rise to this camouflaging coloration, which helps these mice survive in these novel beach habitats. Now, using the same types of approaches that we've been able to use to make links between genes and morphological adaptations, we're now applying to genes and behavioral adaptations. And today I'm going to tell you a story about one of several behaviors that we work on in the lab, again highlighted by this image here, because this mouse is emerging from its freshly excavated burrow. So today I'm going to tell you about burrowing behavior. So to begin, let me start by saying that the field of behavior historically has been described as being largely fractionated. So this was initially described by my friend and colleague Ed Wilson when he argued that behavior was really studied by these two disparate groups. And so on one end of the spectrum, we have folks that would be considered behavioral ecologists who study a wide diversity of behaviors in a wide diversity of organisms and tend to study those behaviors in their natural habitats and ask questions about the ultimate mechanisms giving rise to behavioral variation. So why do these behaviors evolve? On the other end of the spectrum, we have folks that we might consider to be neurobiologists, geneticists, who by contrast focus on a handful of genetic model organisms for very good practical reasons, tend to study behaviors in a nicely controlled laboratory environment, and tend to focus on more mechanistic questions, the proximate mechanisms of how behaviors vary. But what I think is so exciting about this time now is not only is research continuing in these two fields, but the boundaries between these two fields are starting to dissipate. And what that offers then is an opportunity to work at the intersection of these two, and in some sense take the best of both worlds, and thereby ask questions about the molecular, genetic, and neurobiological basis of behavioral evolution, and again, focusing on behaviors that matter to organisms in their natural environments. So to start, what I want to do is just to introduce you to a handful of questions that are not meant to be comprehensive, but to give you a sense of the types of questions that motivate the work I'm going to talk about today. So to start with an age-old question of what are the relative contributions of genetic versus environmental variation to behavioral differences that we observe in nature? And for those traits that do have a genetic basis, what are the genetic changes that underlie those behavioral variants? And it's not just that we want to know the names of those genes or what the precise mutations are. We really want to know how those genetic changes through neural circuits give rise to variation in behavior. And a question that's fascinated me recently is how do those changes have specific effects on certain behaviors, but not affect all behaviors or many behaviors more generally? So to answer these types of questions, we really need to start to make the connections between genes, circuits, and behaviors. So arguably, many of these questions are sort of long-standing questions of broad interest to biologists, so why don't we have more examples of these connections? And I'd like to argue that at least one of the major challenges is simply relative to morphological variation is that quantifying behavior becomes hard. And I want to illustrate this with this sort of simplistic example. Arguably one of the best studied, most stereotyped behaviors is Drosophila courtship. And you'll be able to tell I'm not a Drosophila biologist, but this is how I understand the courtship ritual happens. A male and a female will be in a bottle, they'll orient, they'll start to tap each other, there's some singing going on, licking, they attempt to copulate, and all of this happens before the actual act of copulation. So you can quantify this in an individual to some extent, but now if you start thinking about variation among individuals or even between species, how do you start to measure this? Is it how quickly they orient, the angle at which they orient? Is it the tapping or how vigorously they sing? You can see right away this is much more complicated than measuring, let's say, the length of a femur or body weight or some morphological trait. But what's happening recently, which I think is really exciting, is that there's new advances that are coming online to automate this analysis. Now, while this doesn't necessarily point us to what particular aspects of any behavior are most biologically relevant, at least this allows us to, in a relatively unbiased way, get reliable, repeatable measures of behavior. So I just want to highlight one example of this, which is really the work of Bob Dada and Ryan Adams at Harvard, in which they can take a mouse, a freely moving mouse, put it in a box, use two video cameras to capture its movement. And now what they do is use a computer to put a spline from its nose down to its tail. And you can capture different postures of this mice. And then a computer program will pick out the minimally repeatable units of mouse posture. And you can think of this as a syllable. Now, when you put together these syllables, which I've illustrated in this image here, syllables together can form words. So, for example, a series of postures will be the same as a mouse sniffing, or will give you the readout of a mouse sniffing, and that could be a word. And then you put those words together, and you start to get the body language of the mouse. Okay? So in this way, what's very interesting is you can take this freely moving mouse behavior, and effectively what we're doing is we're translating this behavior into a morphological trait, which is the posture of the mouse. To make this an easier thing to do, as I argued, it's easier to measure morphology than behavior. So while many of these new approaches are coming online, there's another way to use this same trick of taking a behavior and making it more like a measurable morphology that I'm going to tell you about today. And that is, we're going to take advantage of an approach or an idea that was first put forth by Richard Dawkins, who many of you are likely familiar with. in his arguably second most famous book entitled The Extended Phenotype, which was published in 1982. And in this book, he argued the idea of an extended phenotype, meaning that if there are genes that control a behavior, and that behavior results in the production of an animal architecture, and I'll give you examples of that in a minute, then you can treat that animal architecture just like you would treat any other phenotypic trait, and subject it to genetic dissection. So here are just some examples of animal architectures. So here we're looking at a picture of a male bowerbird who performs a behavior which results in the production of this bower. This particular species decorates his bower with blue stuff he finds in the environment. He'll strut back and forth in that bower to attract a female mate. But within this species, the bowers tend to be very similar in size and shape and decoration. In addition, we have the nests built by swallows. The size, the shape of those, where they're positioned in the environment, again, similar within species, different between. Spider webs, another classic example. And then the final example is slightly more complicated because these termite mounds are built not by a single individual, but multiple individuals. But if there are genes that control the behaviors that result in these animal artifacts, what we can measure instead of the behavior are these morphologies. And what Dawkins said now some, 10, 15 years ago, so maybe 35 years ago, I said that nobody had done a genetic study using animal artifacts as the phenotype. I think this is still true, and I think it's still true today. I would admit to disappointment, except that invites the obvious retort of why didn't you get out there and do it yourself then? So lucky for us, Dawkins didn't do this, and so today I'm going to tell you about our efforts. So as I alluded to earlier, the extended phenotype I'm going to tell you about is burrowing. This slide serves to illustrate the idea that burrowing has evolved multiple times in a wide diversity of organisms. So here I'm showing you just a handful of organisms and then more or less realistic depictions of the burrows that they produce. Because it's evolved multiple times, this suggests that it has some functional significance, and we can all start to think about why burrows may be important to an organism in the wild in an ecological context. So it can be important for predator avoidance, for thermal regulation, for those species that meet and mate underground, social interactions, for food storage and growth, just to name a few. But the other reason we got interested in burrows was not only because of its ecological relevance, but also because of some beautiful work done by a quantitative geneticist at the University of Colorado in Boulder, Carol Lynch, who showed in laboratory mice, if you give them the opportunity to burrow in a box filled with dirt, they'll excavate burrows, as indicated by this outline here, and here's an entrance tunnel, that at least some aspects of this burrows that they produced had a heritable component. So here we have the mix between some ecological relevance as well as the possibility that burrows, at least in some species, could be, the shape of those burrows could be heritable. Well, because we're interested primarily in traits that matter to organisms in the wild, instead of working on lab mice, our lab has been developing this particular beast as our model organism. So this is Peromyscus, also referred to as deer mice. To many of you, this looks like a typical mouse, I'm sure. But these guys actually fall outside of the split between our laboratory models, mouse and rat, which maybe are 10 million years. These guys are about 25 million years diverged from them. So this means two things. One, Peromyscus really does represent its own evolutionary lineage. So there are things that we could learn in Peromyscus that we may not learn by just studying mouse and rat. Yet at the same time, they're closely enough related to mouse and rat that we can borrow a lot of those wonderful genetic and molecular tools that have been developed in our model organisms. So like laboratory mice, we can bring these mice into the lab. They breed happily. We can do nicely controlled behavioral experiments. We also are producing a number of genetic and genomic tools like full genome sequences. We're working on transgenesis, et cetera. But what's important to remind you of is that unlike laboratory mice or their wild counterparts, house mice, these mice are not human commensals, and they're found in a wide diversity of habitats. So here's the range of just a single species of deer mouse in North America. So you can find them at the top of the Rockies and the beaches of Florida or Maine. And because they live in such diverse habitats, there's a lot of opportunity for local adaptation, which certainly gives rise to the tremendous amount of both genetic and phenotypic diversity that we observe within and between species. Now today I'm going to start by focusing on just one of the 50-odd species of Peromyscus. So let me introduce you to the main player of today's talk. This is Peromyscus polyanotus, also referred to as the old field mouse. And it's called the old field mouse because you can only catch these mice in old field habitats. And what that means is the habitats are very open, and if shrubs or trees start to form in those areas, the mice will move out. So here are two pictures of our field site. So this is the slightly more glamorous one. This is this wonderful beach habitat that we go to in around February down in Florida to do field work. Here's a slightly less glamorous but equally warm site. This is a burnt peanut field in Alabama. But what classifies these two habitats are that they're open and there's very little vegetative cover. The reason we got interested in this particular species of Peromyscus really goes back to work by this fellow here. This is Francis Sumner, a classic natural historian who spent the bulk of his career driving around North America, trapping mice and describing variation within and among species. And in particular, he did a very nice work with his graduate student. And this image serves to illustrate the convergence between PIs and their graduate students over time. So this is the work, the clothes that they would wear when they would go out and trap mice in the field. We don't even dress this nicely to go to work these days. But Sumner, with his graduate student Carol, in the 1920s published this really interesting natural history observation. And that is about the burrows of these mice. So they were driving around Florida, were trapping these mice, and noticed that these mice throughout their range would build burrows that looked like this. So often in the side of a small hill or rise, you would see an open entrance tunnel. And if it's an active burrow, there would be this plume of freshly excavated soil on the outside. Then there would be a long entrance tunnel, sometimes plugged with a little bit of loose dirt, that reached down to a nest chamber. And then a secondary tunnel that importantly doesn't penetrate the surface, but radiates up towards it. So next what I want to do is tell you about how we catch these mice in the field, which is going to illustrate two important points about this burrow architecture. Okay, so as I mentioned, around February, we'll go down to the Florida coast. We'll look for these open field habitats. We'll drive around, and once in this open field habitats, we'll look for something like this, where here's that entrance tunnel, here's a plume of freshly excavated soil, we have an active burrow. The next thing we do is get a graduate student and ask them to lie on their belly, face first to the entrance tunnel. And that's what Jesse Weber is doing here. Now, while it looks like the rest of us are laughing at him, we actually are going to play a really important role. So what we do is we take tubing, and we just take that plastic tubing and we'll weave it down that entrance tunnel. So that's what Jesse is doing till he hits the nest chamber. And then once he hits the nest chamber, he blows into that tubing, disturbs the mice inside. And then what's going to happen is those mice are going to come popping out of that secondary tunnel, and we literally jump on them with the goal of catching the mice that come out of that burrow. Okay, so I'm telling you this for two reasons. The first is that secondary tunnel is actually used as an escape hatch, such that if something comes in the entrance, the mice have a way out the back door. But second, these mice are very fast. So the lab mice that many of you work with are fast, right? They get out of the cage, you have to chase it down, they're fast. These mice are 10 times faster. And the only reason we have any hope of jumping on them as they come out of this escape hatch, which remember, you can't see the hole, it doesn't penetrate the surface till they come out, is that the size and shape of this burrow is so stereotyped, we can predict with great accuracy where that escape tunnel will be. So we're standing at the ready because we know where that hole is going to be. So you may be wondering why they produce this sort of complex burrow shape, and the answer, I think, may be obvious, and that is that one of their major predators are snakes. So if a snake comes in, for example, this very obvious entrance tunnel, it's a good idea to have a backdoor escape hatch. The other thing that we know about these burrows, in addition to this natural history observation, comes from work by Wally Dawson. So Wally Dawson is thought of as the father of Peromyscus biology and is actually the founder of the Peromyscus Genetic Stock Center. And what he did was he brought these mice in from the wild and brought them into the lab, put them in a box of dirt, and asked, will they recapitulate this natural behavior in the lab environment? And in fact, what he found is that they will, and they will still produce this entrance tunnel and escape patch. So what we decided to do, now armed with molecular tools, is ask first, do they still recapitulate this burrowing behavior now some 40 years later, and can we start to understand its genetic basis? So here is our, one of our rooms in our mouse facilities where we can breed the mice, and then we've built these 10 boxes. Each box is quite large. We refer to them as our phenodomes. In these large phenodomes, there's about 1.5 tons of dirt. We give them this slope because this will elicit a burrowing behavior in the center of the slope as opposed to one of the corners. Each assay involves taking a single mouse, putting in the box, leaving it in there for 2 activity periods or 2 days, completely undisturbed. Then we come back and take the mouse out. And then to measure its behavior, here's where we get really high tech. We use what we call phenofoam. Phenofoam is the stuff you would buy at Home Depot or Lowe's. That's an insulating foam. You squirt it into a hole. It will expand to fill that space and then harden. So what we can do then is produce a cast of the tunnel, or, in other words, an extended phenotype that captures the behavior that that mouse performed over the previous 2 nights. So let me just show you how this works in the lab. It's the same thing we do in the field. So here's Jesse weaving that tubing down the entrance tunnel. Out popped the mouse out of its escape tunnel. You didn't see that escape tunnel before the mouse popped out. Then we trap the mouse, take the mouse out of the box, and use the same sort of approach to fill it with our phenofoam. And you'll see the foams come squirting out on this end, so it goes underneath in this tunnel. We let this harden then for a few hours, and here's where having eager Harvard undergraduates comes in. They help us dig these out, get their hands literally dirty, and start to capture this data. So in this case, each mouse we test 3 times, and the data I'm going to show you today comes from the best burrow that the mouse produces, which is most often the 3rd of those burrows. If we did it a 4th day, we don't see any differences past the 3rd day. And importantly, these are mice that we put in these boxes that they themselves have never seen dirt. Their grandparents have never seen dirt, and some haven't seen dirt for up to 50 generations. So using this approach, we actually run into, I would say, a classic big data problem, because this literally results in mountains of data. So we have a data storage problem. We have now filled the entire attic of the Museum of Comparative Zoology. Here's an undergraduate for scale, right? So what we have now is we've gone through thousands of mice, each one tested 3 times, so you can imagine how this has quickly got out of hand. So we can go to measure the mouse behavior or its extended phenotype. We can go in with a ruler and already start to see big differences. But we've become, over the years, more sophisticated and collaborated with applied mathematicians who now we can take video images, recreate these burrow casts in 3D, use a computer algorithm to measure all sorts of things about this burrow, its curviness, its volume, its length, its the number of tunnels, et cetera. It picks out the nest chamber, for example. So today I'm going to focus on just 2 of the very simplest measures. So we're going to talk about the length of the burrow. So here's a short burrow relative to a long burrow. And we're going to talk about the presence or absence of the escape tunnel. So here's an escape patch. Here's a burrow without an escape patch. Okay, so when we started on this project, one of the first questions we wanted to know is this behavior unique to this species? What about the other species in this group? So to address that question, we simply repeated the experiment I told you about earlier, taking a mouse, putting it in one of our phenodomes, now with a diversity of species that have been acclimated to the lab, which represent the diversity across Peromyscus. So the first thing I want to tell you about is that there's a lot of variation among species. So here I'm showing you 2 simple measures, the frequency of burrowing in green and the total burrow length in gray. So the first thing to note is that there's a lot of variation among these species in both the length of burrow they produce and the frequency in which they produce them. The second thing you'll notice is those 2 measures are highly correlated. Species that tend to build burrows more frequently, when they do build a burrow, build a longer burrow. And 3, that there's some species that don't build any burrows in our boxes. And we were worried about this at first until we went back to the natural history literature, and species like Peromyscus ceremicus, which is a rock-dwelling mouse, doesn't build burrows in the wild either. It lives in rocky crevices. So we think we're capturing ecologically relevant differences. But as I told you, the length is interesting, but really the shape is also very interesting. So here in very cartoon-like form, I'm highlighting the typical shape of each of the burrows that these species make. So we have no burrows. Many make these little sock-shaped burrows just a few inches underground. Here's our big Polynotus-like burrow. This other species, Aztecus, which builds burrows that are just as long and just as frequent as Polynotus, they have a long entrance tunnel, but importantly, they are missing that escape tunnel. So the escape tunnel appears unique to Polynotus. I think this data becomes even more interesting when we look at it in a phylogenetic context. So here are the relationships among those species based on genetic data. And we can learn things like the escape tunnel is really a unique trait or derived trait, meaning when we study the escape tunnel, we're learning about the increase in complexity, behavioral complexity, as opposed to the loss of behavioral complexity. And the other thing that this phylogeny tells us is that even closely related species like Maniculatus and Polynotus, which are sister species, all the good species in the wild, when we put them in the lab and don't give them a choice, will happily mate and produce viable and fertile offspring, allows us to harness the power of forward genetics to start to ask about the genetic differences that give rise to little burrowers versus big burrowers. Before we did that, however, we wanted to really try our best to make sure that there was a genetic component to this. So we did first a cross-fostering experiment, where we took the offspring of Maniculatus, had them raised by Polynotus parents, and Polynotus pups, when they were born, were cross-fostered by Maniculatus parents. So these are the 2 species I'm going to focus on today, just to see if there was any postnatal effects or if there was any learning from the parents. And the very simple answer is that there was no measurable effect of postnatal environment. So Maniculatus, the little burrowers, whether they were raised by their own parents or the big burrowing parents, always built little burrows. Note the change in scale here. These guys always built relatively large burrows. Okay? This was another piece of evidence that got us excited about maybe there's a good, strong genetic component. And at least bolstered our confidence enough to take the next step, which is to start to do crosses. So in the lab, we had Polynotus, our big burrows, Maniculatus, our little burrowers, and we made F1 hybrids, and then asked, what kind of burrows do F1 hybrids make? So this is where a lot of betting went on in the lab. Is it going to build a big burrow, a small burrow, something in between? And it turns out that these guys build perfectly complex long burrows, indistinguishable from Polynotus. So here's our F1 hybrids. Polynotus make big burrows, Maniculatus little burrows. But even more striking is the frequency of the escape tunnel. Maniculatus never build escape tunnels. Polynotus always do, and F1 hybrids always do. So this suggests that the complex burrow, the underlying genetics, seem to be dominant in terms of the complex burrow or the Polynotus-like alleles. This then led to our next step. We took the F1 hybrids, back-crossed them to Maniculatus, and asked, what do the next generation of now recombinant hybrids do? So I'd like to show you just the raw data because I think it really gives you a sense of the segregation patterns. So here's Maniculatus, our parents, little burrows, Polynotus, big burrows. So we're considering just the length of the burrows here. F1 hybrids tend to build long burrows like Polynotus. And then when we look at it, take these guys and mate them to these guys, here is our distribution among our back-cross individuals. And what you can see immediately is that this is not like Mendel's peas. There's continuous distribution, so it's not consistent with a single genetic effect, maybe not surprisingly. But interesting to us is that there were still mice in this back-cross generation that only have a quarter Polynotus alleles that still built long Polynotus-like burrows. And you can actually use this distribution in this back-cross to, I would say, guesstimate how many genes are involved. And we guesstimated that there were a handful, about 3 to 5. So this, as a geneticist, was great because one gene may be a little boring, 10 genes, you're getting a little intractable, and we're somewhere in between. But again, this is just length of the burrows. What about the shape of the burrows? So if we again look at the back-cross individuals and start to ask what types of shapes do these back-cross mice build, some build little Polynotus-like burrows, some build, little Maniculatus-like burrows, some the big Polynotus-like burrows. but we got mice that also built long burrows without an escape or short burrows with an escape. So this right away told us we could genetically dissociate the length of the tunnel from the presence or absence of the escape tunnel. And then we went one step further and asked, if we just count up the number of back-cross mice that built no escape tunnel with those that did build an escape tunnel, what we got was sort of a geneticist dream, as close as you're ever going to get with real data to a one-to-one pattern. And that is consistent with the idea that the presence or absence of the escape tunnel may be controlled by one genetic locus. Importantly, there are other genetic explanations that can explain this data, but of course the most enticing one is that there's a single gene. So the next step, of course, is that now that we know something about the genetic inheritance pattern, is to ask what are the genes that are underlying these differences. So to do that, we used a quantitative genetic mapping approach. So what we did is for all those back-cross individuals, we've now measured their burrowing behavior. Now we're going to next genotype them at multiple markers across the genome. And this is not trivial given that we don't have a Peromyscus SNP chip. So we developed a new genotyping by sequencing method, which allows us to quickly assay multiple markers across the genome. I'm not going to go into details with that. But to suffice it to say, we could genotype them. Each of these dashes represents a polymorphic SNP. And then ask, what's the correlation between genotype and phenotype? In other words, are there any particular genotypes that are predictive of the behavior or the burrowing behavior of that mouse? And here I'm showing you the four chromosomes that show an association. So here's our LOD score, that association between genotype and phenotype, and our genome-wide significance threshold. In black, I'm showing you entrance tunnel, and what you can see is there's 3 regions of the genome that are associated with entrance tunnel. Now remember, I told you the length of the tunnel had this continuous distribution, and we guesstimated about 3 to 5 genes involved. So this is consistent with that. Second, there's only one region of the genome that shows a statistical association with the presence or absence of the escape tunnel, consistent with the data that I showed you that had this tantalizingly Mendelian inheritance pattern. And then the third thing this data shows you is that the regions of the genome associated with entrance tunnel length are different from that associated with escape tunnel length. So again, consistent with the idea that you could genetically dissociate these two because the causal loci are on different chromosomes. So now, depending on if you're a human geneticist or a Drosophila geneticist, these LOD scores may be more or less convincing to you. So the way that looking at the data that really convinced me we were onto something is looking at it in this way. So here, if we now consider the genotype just at the marker that showed the highest association, remembering that polyenotis is dominant, if we have a single polyenotis allele compared to mice that have 2 maniculatus little digging alleles at this particular locus on chromosome 1, on average, the mice produce burrows that are 3 centimeters longer. If you do that same experiment with now this marker on chromosome 2, the mice build burrows that are on average 3 centimeters longer. If you do that again with this data here, on average 3 centimeters longer. This is not Photoshop, this is real data, and we had no a priori expectation of why these appear to have the same effect size on burrowing, a 3 centimeter effect, and why when you add these all together and you look at all 3 QTL together, they largely recapitulate the difference between the two in an additive fashion, I think is really interesting. Similarly, if we look at this marker here at the end of chromosome 5, we see that there's a higher propensity to dig an escape tunnel. Okay. So now we have these QTL, and the big challenge of QTL mapping is going from a whole list of, let's say, 100 candidate genes in a QTL peak to finding the causal gene. And this is what we've been spending the bulk of the last several years trying to do. So the first way we're attempting to do this, I'm going to focus on this peak here on chromosome 2, which has a repeatable, reliable, and a large effect on burrowing. And that is, we took the best burrowers from the back-cross generation, back-crossed them to maniculatus, then took the best burrowers from that generation, and we did that 7 times. And so in doing so, what that does is it narrows your QTL peak. And so our peak is now getting smaller, and our list of candidate genes are getting smaller. And depending on what approach you use to mapping, you can make these peaks look really small. But today I'm going to tell you just about our most conservative estimate, and really focusing just on our first back-cross interval. So we're highly confident that this region contains a causal gene associated with length of the burrow tunnel. And in this region, there are about 54 genes. At the time, we didn't have the genome sequence of Peromyscus, but they're highly synthetic. And it turns out these same 50-odd genes are in the same region in the Peromyscus genome. So how do we go from 50 genes down to a causal gene or a few genes? So here, I'm going to tell you next our thinking through this. First thing to tell you is that of those 50 genes, about half of them are olfactory receptors. Now, I'm not meaning to say that olfactory receptors are definitely not involved in burrowing behavior, but for the moment we push them to the side as being not our best candidate genes. Okay, I know somebody's going to argue with me about that, but we'll deal with that later. So then we're left with about 25 genes, and we ask the simple question of those genes, which ones are expressed in the brain? And it turns out 8 of them are expressed in the brain. Again, we may be missing things, but this was just our way to prioritize candidates. We next asked which of those brain-expressed genes in our QTL peak show differential expression between our 2 species. Now we're down to 3 genes. And then we got really excited about one gene in particular, and that's this M5 receptor here in the middle of our QTL peak. And I'll tell you why. So as I mentioned, it shows differential expression between our 2 species. So here's Polyanotus, here's Maniculatus. This is early data with very small sample sizes. So 1, it shows differential expression. 2, if we look at the expression of the F1 hybrid, it shows expression similar to Polyanotus. Remember, it builds burrows similar to Polyanotus. And then 3rd, if we look at allele-specific expression in F1 hybrids, so in an F1 hybrid that has both alleles, how are those alleles expressed differently in the same genetic environment? It's the the Polyanotus allele that shows higher expression, consistent with this gene having a cis-regulatory mutation instead of a transacting effect. Okay, so all of this data got us super excited about this gene. This is a great candidate. So next what we wanted to do is actually try to perturb this gene and see if it has an effect on burrowing behavior. But what we realized is that our wonderful extended phenotype approach, which got us out of having to measure mouse behavior, actually now we might have to bite the bullet and look at mouse behavior itself. But remember, this is a behavior that happens at night, in the dark, and underground. So how are we going to measure this behavior? So to do this, I had a postdoc, Brant Peterson, who came up with a wonderful idea, inspired by at least some of us in our youth had ant farms. So we made the equivalent ant farm for a mouse. So this is a 2-dimensional slice of one of our big boxes, which now we can watch what the mouse is doing, videotaping it with this plexiglass side under dark conditions. This is in daylight, just to give you an example. And all of a sudden, we can start to see what the mouse is doing itself to produce this burrow. And we learned all sorts of neat things, like it goes down here, and to start to build its escape tunnel, it flips on its back and digs up. We've learned about how it carves out its nest chamber, how it goes out and about and explores its environment, takes a break, comes back in and burrows. So all of a sudden, we got so excited about mouse behavior itself. And then we realized something, that if we were going to measure each mouse again 3 times for 8 hours, we could have a whole army of Harvard undergraduates, eager as can be, but we were never going to be able to watch that much video, much less how do we score this video? Is it how many times it flips over upside down, how many paw strokes it does? So again, we were stuck with this problem that we started with of how do we measure behavior. So here's where we went and automated this behavior. So what Brant did is using computer vision, now can track the mouse moving in this ant farm in an automated fashion. So that white blur is the mouse that we're tracking. And here on the bottom, this is obviously sped up. It's 8-hour period over 900-fold. And we're measuring 2 things. We're measuring this pileup of excavated soil that's coming out of the burrow, which is highly correlated with the amount of visible burrow down below. And so we can start to ask, what is this mouse doing now in an automated fashion? But I think this behavior is much more interesting when we contrast it with what the other species, the little digging Maniculatus, is doing. Here in the exact same setup, we're tracking the behavior of that Maniculatus, and you can see it's definitely active. It's running around like crazy. And this particular mouse has built itself this little ramp, so sometimes it can do these back flips. Basically, it runs around. If you're getting bored, that's a little bit of the point, right? This guy is doing everything but burrowing. It runs around, does some backflips, runs around some more. And then a few hours before the lights come on, it quickly builds this burrow, a little one over here too, and then basically sits in the burrow for the rest of the trial. So now what we can do is use this automated way of measuring behavior to ask basic questions about what's different in the mouse behavior itself over hundreds of thousands of trials that give rise to the differences in the burrows they produce. So we started with some very simplistic questions. For example, do they dig at different rates? Does Polyanotus dig a bigger burrow because it just digs faster? And indeed, Polyanotus, the big diggers, do dig faster, but this only explains a small percentage of the difference in burrow length. So they dig a little bit faster. Okay? But what you probably already gleaned from those videos is the big difference in digging comes from the amount of time they spend digging. So Polyanotus spends significantly more time digging compared to Maniculatus, and this really explains a lot of the difference in burrow length. So now what we can do is focus in on the time spent digging. And to us, what this means in a neurobiological context is the results implicate, and I'm going to use the word motivation in a very loose way, so I apologize to anybody who thinks very carefully about motivation and what it means, but for lack of a better word, we were implicating a motivational difference here rather than, let's say, a mechanical difference. The other thing that you've probably gleaned from the video is it's not just the duration of time that they build burrows, but it's when they start building a burrow. So Polyanotus almost immediately start building a burrow within the first hour or two they're put in the box. But by contrast, Maniculatus is running around and starts to build burrows only just before the lights come on. So we refer to these as our eager beavers and our procrastinator mice. So again, this is consistent with what one may consider a motivational difference. So one of the things that we're doing now is instead of mapping the genetic changes responsible for the extended phenotype, we're starting to map differences in the behaviors of the mouse themselves and ask, do those overlap with any of the differences that we see in the burrows? So I just quickly want to show you some brand new data, sort of hot off the presses, in which we're looking at this difference that I just highlighted in the latency to build a burrow. And here what we see in our genetic map is there's a really strong peak with chromosome 1. As a reminder, this was one of the QTL peaks that came out when we looked at the length of the burrow. And you can see here that we have this really nice, relatively small region associated with when they start digging that overlaps with the burrow QTL. In addition, we see that it's in the direction we expect. That is, if you're homozygous for Maniculatus at this particular peak on this chromosome, you tend to build burrows later compared to Polyanotus, and the average is about 45 minutes later. And in addition, those F1 hybrids, or individuals that are heterozygous at this particular locus, are dominant for the Polyanotus behavior. They also build burrows early. So now what we're doing is complementing our mapping of the extended phenotype with the mapping of behaviors. So we really got excited about this idea that there's a difference in sort of their motivation to dig, including this phenotype of how early they dig in the night. So I showed you this data already, that there's a difference in how long they dig when they dig. There's also a difference of when they dig during the night of their trial. These data I showed you as well. There's a difference between these two species in how frequently they dig and when they dig, how long their burrows are. So Polyanotus starts digging earlier in the night. They dig for a longer time. They dig burrows more frequently. And then in addition, they also dig burrows earlier in ontogeny. So if we take our mice, which just for context, we wean around 23 days of age, the first Polyanotus mice will start digging burrows at only 17 days of age. So think about this in the mouse cotton. This is a teeny weeny little mouse. We couldn't do 15 days of age because they can't survive that long without their mom. But they're already building burrows, and they're building complex burrows, complete with an entrance tunnel and an escape tunnel. It's just miniaturized. By contrast, Maniculatus doesn't build its first burrows till 10 days later, at 27 days of age. And when we start to look at the correlation between the type of burrow that an adult builds and when they start building in ontogeny, these are correlated. So the ones that start building earlier are Polyanotus-like in terms of their ontogeny, also build larger burrows as adults, suggesting that these handful of genetic loci may affect many aspects of behavior, but all associated with burrowing. Okay, so we want to end by taking you back to this gene that I told you about. We were so excited about its expression pattern, but needed to look at the mouse behavior before we started to test this. So let me just set expectations. We don't yet have the data that definitively implicates this gene, but what I want to show you is some tantalizing evidence that suggests this is a good candidate. Okay, so what do we know about this mysterious M5 gene from mice and humans? So it's a muscarinic acetylcholine receptor. It's part of a family. This is the 5th member, and of course is the least well-studied of the bunch. It's expressed in dopamine-containing neurons in the basal ganglia of mice, in particular the VTA, which is associated with reward circuitry, and the substantia nigra, which is associated with, among other things, motor control, spatial learning, and addiction. What we know from knockout studies in mice and GWAS studies in humans is that there's a morphine addiction phenotype and a nicotine addiction phenotype associated with this locus. So what's clear is that M5 modulates dopamine release in the brain's reward center, and I have to say, at watching these mice dig, it's hard not to imagine that they're addicted to burrowing. I use that very loosely. Okay, so the first question we asked is, in Peromyscus, is this gene expressed sort of in the right places? In the same places as in mus. And so we did some very simple staining with M5 and tyrosine hydroxylase, and it does seem like chrome 5 is expressed in dopaminergic neurons, as in mus and Peromyscus. So it's expressed in the same place. Now we can ask, when is it expressed? Is it expressed at the right time? And here we could take advantage of this difference in the appearance of first burrows between these two species. And what you can see is that in Polyanotus that start digging early, the gene comes on earlier and is expressed at higher levels during this key time points. So we got excited. It's expressed in an interesting place, expressed differentially at the right time. So where it's correlated to expression, to the burrowing behavior itself. And now we've started to poke at this gene, initially with a blunt tool, pharmacology. So remember, it's an acetylcholine receptor. So what we can do is give mice acetylcholine esterase inhibitor, in other words, increase the amount of acetylcholine. And what we can see is that there's a change in the length of the burrows that they produce. So it's correlated with increased burrowing behavior. So here is where we are now. We're working on transgenesis and using viral vectors to affect the expression of this gene and acetylcholine. So we're left with the question of, is this the right gene? And I think the answer is maybe. And it's not just that we want to know what this gene is, because I think finding the gene just represents the first step. Next, we want to know is how this gene actually works to produce variation in burrowing. And what I'm particularly interested in is that it seems like the differences between these mice are largely confined in this context to burrowing. So if we look at other motor-related activities, so here's just one example of wheel running. It's not just that Polyanotus are more active all the time. If anything, Maniculatus runs longer in a wheel for a larger amount of time. So the differences that we see in activity or motivated behavior seems to be associated just with burrowing. So is this the right gene? How does it work? And in particular, how do we make changes at the genetic level that have such behavioral specificity? So let me tell you quickly how we're going about answering these questions. So we're doing more mapping as a geneticist would. This is a little bit slow. We continue to now map the behaviors I told you about. We're complementing this, now taking a multi-pronged approach with comparisons of gene expression. We're doing this at whole brain level, but also using IEGs to identify regions of the brain that are active during burrowing. So we can do comparisons between species, between brain regions, across ontogeny, before and after digging, or allele-specific expression to try to narrow in on candidate genes. And once we have a good list of candidate genes, we're working to use viral injections and CRISPR, of course, to try to see if we can get little diggers to dig big burrows. And then finally, we're doing a series of manipulative experiments, which I think gets to this point of looking at behavior itself can be really informative. So asking questions like, if you give the big digging species a burrow that's halfway already made, does it build a burrow that's the full distance, or does it add, let's say, 10 centimeters onto its burrow? So is it interested in the final product of the burrow, or is it interested in digging for a certain length of time itself, or giving a certain amount of effort? And I think these types of experiments will also help us interpret the molecular data. So our ultimate goal is to identify specific genes that confer ecologically relevant differences in behavior. And we do this for two reasons. Finding the gene gives us a handle in which to ask mechanistic questions. So where are these genes that affect behavior expressed? What are the neural circuits that are involved, and how do they act to have such specific behavioral effects? But trained as an evolutionary biologist, we're also interested in using the genetic information to understand its evolutionary history of this behavior. Where and when did the derived allele arise? Is there evidence of selection driving it? Does the same gene contribute to digging behaviors, the same allele in digging behaviors in other species? So to do this, we're taking the best of behavioral ecology and neurobiology and genetics to try to make these connections. So before I end, I want to just tell you very quickly in the last 2 minutes about other behaviors that differ between these species. Because I've spent the bulk of today telling you about what we know about burrowing, but our lab works on a number of different behaviors, including differences in exploratory behaviors. Polyanotis, this open field specialist, spends significantly more time in the open arms of an elevated plasmase, which is consistent with it having evolved in an open habitat. These two species also differ in mating system. Whereas most mammals, sorry to say, are highly promiscuous, one of the rare exceptions is Polyanotis, which is highly monogamous, both genetically and socially, which has an effect on their parental behavior. We've now identified a gene contributing to differences in parental behavior. Because they have different mating systems, there's differences in the amount of sexual selection. In promiscuous species, there's a high level of competition, for example, sperm competition. And this results in differences in sperm morphology and sperm swimming performance. So promiscuous sperms swim very fast. They're in a race to the egg. Monogamous sperm get to the egg but take their sweet time. And this is in part driven by morphological differences in the midpiece, which we've identified a gene that contributes to that, which then has an effect on the likelihood in which a male will sire offspring. So in addition to being interesting in terms of their burrowing behavior, we're also interested in finding genes that contribute to the suite of other behaviors to try to get a bigger sense of how genes through neural circuits contribute to behavior. So let me end by thanking the following people. Jesse Weber is the graduate student that took on this project. He said, morphology is boring, give me something tough. We handed him a behavior and he ran with it. Brant Peterson is responsible for all that automated video analysis in the ant farms, which we fondly refer to as the Brant farms. Hilary Metz has done all the hard work doing the genetic mapping. Nicole Bedford has done a lot of the behavioral mapping of the behavioral work and is also out in the field looking at burrowing behavior in the context of wild mice in their natural habitats. Stuff that I didn't have time to tell you about today. This work, as I mentioned, also included a wide number of undergraduates, and literally everybody in our lab has gotten their hands dirty. And with that, let me answer any questions. Thank you.
Thanks for a fascinating story. My goodness, I'm sure there are questions, so please use the microphones. I see people are doing that already. Please start here.
In terms of animal artifacts, it should note that Darwin actually spoke about bees and the honeycombs, and he made it explicit. He was saying, how are they making these excellent angles and things? That sticks in my mind.
Yes.
So anybody who reads Origin of Species should remember that.
Yes.
And with respect to what you were just talking about, sperm, there are a bunch of papers now about microRNA. Changes in microRNA are a heritable characteristic in trauma in laboratory animals. Now, it's not just one paper, it's several. They kind of reiterate the results of the others, but that might be of interest to you to trace down for the implications in humans and psychiatric, et cetera.
Yep, absolutely. I think those are both great points. There's a whole chapter devoted to honeybees and the origin of species, which I agree everybody should read as a biologist. That's sort of one of the Bibles of the field. And then the second thing, I think I definitely agree that we tend to think of, and the way we teach this too is, you think of a mutation, you think of a single SNP and it changes. And I think there's a lot of interest in understanding what the actual molecular changes are and if they involve structural rearrangements, if they involve regulatory changes or structural changes to proteins or microRNAs, and how that all sort of fits in. And I think we're just starting to get a handle on sort of the fodder for evolutionary change, so to speak. And I don't think we have any big picture yet. We're still gathering that data, and it's going to be really interesting.
Thank you.
Thanks. Over here.
That was super. Thank you very much. I was wondering if you have PPO in the this ant chamber, and in the middle of the night you turn the light on. Can it stop, or does it keep going?
That's a great question. We haven't done that experiment. The experiment I can tell you, which is a good lesson I think for graduate students, it's one of what I call an accidental experiment. We had maniculatus, the little diggers, were being run in the ant farm. And so we take videos of them, and then that video gets fed to a program that then will quantify all these differences in behavior. And that program takes about 2 weeks to run. So what happened is somehow, and we're still not sure how, the lights in our burrowing room were turning on earlier, about 10 to 15 minutes every day. And we didn't catch this till 2 weeks later because my students are working really hard, but nobody's in there at 4 in the morning when the lights come on. And so we're watching the videos, and then all of a sudden the lights came on too early. That's weird. The next day, oh, now they're on even earlier. So we could backtrack. So we had about 2 weeks of data this was happening. And the mice shifted when they burrowed. These are the ones that are the procrastinators, to dig earlier. So there's clearly, at least in maniculatus, seems to be a clock-like component to that. And my guess is that Polyanotus, the eager beavers that dig right away, just override that, what was probably an ancestral when to dig component, because they just want to dig. They see dirt and they just go for it. That doesn't directly address your question, but I think there are some neat experiments we could do to sort of look at the circadian component to burrowing and to test whether that exists in Polyanotus and it's just being overridden.
It might just get at the motivation if they can't stop.
Oh yeah, in whatever conditions, yep. I think that's a great question.
Let me ask you about CHRM5. I bet I'm not the only person who was sitting in the audience quickly pulling out our iPhones to look that up before you told us what it was and seeing, oh my goodness, it's cholinergic muscarinic receptor. That sounds like a pretty good candidate. Now you showed the expression pattern in the brain of the burrower versus the not so much, and it looked like there was a pretty enormous quantitative difference in the gene expression, almost as if there had been some activating derived allele, perhaps.
Yep.
So one immediately wants to know, what does the sequence look like? Is there something there that you might consider a smoking gun?
Oh, okay. That's a great question because I didn't show you any of this data. So initially when we thought this was a great candidate, and what we did for all of the genes in our QTL-peak is just say, okay, are there any differences in the coding region? Because that's something you could do easily bioinformatically. What's harder is to interpret those. But when we got the expression data of this gene and we started thinking about this gene, it looked really exciting. We said, okay, let's go back, look at the coding sequence. And one of the things we saw was that there were 2 mutations in this gene that were unique to Polyanotus. And what I mean by unique is that it's not just unique compared to maniculatus, but across all mammals. So all other mammals, I can't remember the exact residues, had one base at both of those sites, and Polyanotus had 2 unique ones. And one of them is a site that's associated with the disulfide bridge. And this is a G protein coupled receptor. We started getting excited, like, oh, maybe this is it. And so we did some very simple pharmacology experiments. We expressed the 2 alleles with and without the 2 mutations in cell culture and looked if they behaved differently when I can't remember now what we activated it with. There were no measurable effects. So what that means is that it doesn't mean that these amino acid changes are not important. It just means that we may not, if they are important, we may not have the right conditions. And then we started thinking, should we spend the time, we came to this point, and we're sort of at this point still, is how much time do you go down the road of a single candidate gene and spend going after that, like, so it looks like a smoking gun, versus let's just systematically map in a really boring, slow way, narrow our list, be more sure of those candidates, take these other approaches like looking at gene expression and so forth. So it's still on our list.
So you haven't looked at the promoter.
So these 2 species, the promoter is tough because these 2 species are separated by about 1 million years, which means that every 500 base pairs there's a mutation, sometimes much more frequently. So there are mutations everywhere. So it certainly won't be as simple as let's just compare them and here's the one mutation. But there was nothing obvious in the promoter. There are changes, but not anything that you would jump up and down about, which again is, tells you be cautious, maybe not.
Of course. Over here.
Just a simple question, but you alluded to this when you talked about the snake coming into the burrow of the animal that actually makes the escape. But what is the difference in the actual natural predators between the 2 species?
Yep.
And do you think that plays a role in why one makes an escape hatch and one doesn't?
Doesn't. So the predator composition or community is really hard to quantify, in part because Maniculatus is that widespread species that's basically all over North America, so it's going to be highly variable wherever you are. Polynotus is restricted to the southeast. We know something, their main predators are things like mammalian carnivores, visually hunting owls, herons, and hawks, and snakes. I wouldn't say necessarily that there are more snakes in the south than in the Maniculatus range, because I think the Maniculatus range is so variable, and some places will have more and some less. The thing that I would guess, waving my hands purposefully, is that remember the Polynotus lives in open fields. They are only in open fields. If there's a predator around, or they're just, they have nowhere to hide or run or anything, their burrow is it. So I think that there is this prioritization of building a burrow is my first priority. And I'm going to do that first, and I'm maybe going to build several burrows, because if I go out the escape tunnel, I didn't mention this in the field, if you see the escape tunnel, you'll never find a mouse in there. They abandon that, and that makes sense, because now your predators can come in two ways. Their secret escape plan is foiled. Whereas Maniculatus, which lives in dense fields and forests, can run under a rock, run up a tree, etc. And remember, Maniculatus is the promiscuous one, so its priority may be running around to find mates, and I'll burrow when I have to, but I don't really have to until it's going to be daylight, whereas Polynotus has a different set of priorities. So that's how we're starting to think about it, that it's maybe driven by the environmental differences plus the mating system differences, maybe the predator communities, et cetera, that it's much more complex, and this is the one agent of natural selection that's driving it.
Terrific.
It's a great question.
Well, please, we are all invited to a reception in the medical library with cookies and coffee. But meanwhile, please, let's thank our speaker one more time for a terrific presentation.